Welcome everyone. Here is another question from GATE PYQ series. The question is, DNA in a 1 centimeter long chromatin contains 5 into 10 raised to 9 base pairs. The fold compaction of this DNA within the chromatin. So as we all know, chromatin is a complex of DNA and proteins, and these proteins are either histone-like or non-histone proteins. So as you can see here, this is the DNA and this is associated with histone proteins as well as non-histone proteins. As you can see here, the DNA is wrapped around the histone protein, and the fundamental subunit of chromatin is nucleosome. So this is a 11 nanometer structure, and this consists of segment of DNA. So this is the DNA, segment of DNA which is wrapped around this octameric histone protein. Now the function of the chromatin is to package DNA into more compact and denser structures so that it can fit inside the nucleus. So keeping this information in mind, what we need to do, we need to calculate the fold compaction. So first we need to calculate the length of this DNA when it is not associating with any of the proteins, because it's given the length of the chromatin is 1 centimeter, and this corresponds to 5 into 10 raised to 9 base pairs. Since we need to calculate the fold compaction, we need to calculate the length corresponding from this number when the DNA is not associated with the proteins. So as we all know, the distance between adjacent base pair is 0.34 nanometer. So 5 into 10 raised to 9 base pair will correspond to 5 into 10 raised to 9 into 0.34 nanometer, and this is going to be 1.7 into 10 raised to 9 nanometer. I can convert it into meter, and we all know 1 meter equal to 10 raised to 9 nanometer. So this is going to be 1.7 meter. So what we need to understand is, without any association with the proteins, this is 1.7 meter. And when it gets associated with the proteins, it becomes 1 centimeter, which corresponds to 0.01 meter. We'll keep the unit same, 1.7 meter divided by 0.01 meter. And the fold compaction, we are going to get 170. Now, we need to get the concept right here is this much fold compaction is taking place, which is a big number. As I said, the function of the chromatin is to package DNA into more compact, denser structures, and that's what it is doing here. So this was without protein association, and this was the chromatin, where the DNA was associated with the proteins. I hope this answer is clear to each and every one of you. If you do have any doubt, feel free to comment. Thanks for watching the video.